For the first time in the history of humanity, computing technology is unlocking the secrets of millions of years of evolution. We can easily extract DNA sequences from nearly any organism, and the four-lettered nature of the genetic code makes it simple to turn this biological information into a digital file. If we can find a way to effectively interpret this data, we will gain deep insight into the origins of life itself. My name is Peter Lazorchak, and for the last 10 weeks I joined Jeffrey Fader's lab as a part of Notre Dame's Data-Intensive Scientific Computing REU. During this time, I analyzed big genomic data to understand how and why species change over time. An important contributor to evolution is speciation, or the splitting of one species into two new ones. There are already lots of programs that measure speciation, but a new one called Entropy was developed to take sequencing error into account when performing its calculations. You see, the older programs worked, but they made the unrealistic assumption that all of the input data was perfect, which is almost never true in real-world computing. So why doesn't every biology lab already use Entropy? Unfortunately, it was not designed with the user in mind, so to say it's difficult to use would be an understatement. This summer, I developed a suite of modules to make Entropy's technology more accessible to laboratories around the world. I even modified some of the source code itself to enable compatibility with other already popular tools. Now, it doesn't take a computer scientist to run Entropy. Generating high-quality graphs from raw sequencing data only takes a few easy steps. Armed with my extensions and alterations, I ran some of my lab's own data through Entropy, which combines several populations of Ragoulides flies from the central and eastern United States. These flies are particularly interesting because their speciation is driven by different fruit on which they mate. There's no physical separation of diverging populations. Over time, fewer and fewer flies interbreed with those preferring other fruits, which leads to differences in the flies themselves. My analysis included flies with three distinct host fruits: apple, hawthorn, and flowering dogwood. Here are Entropy's output graphs. Each bar represents an individual, and each color corresponds to a population. So we can see quite clearly that Entropy distinguished between dogwood and others based on the sharp transition between red and gray bars. However, when I told Entropy to look for three populations, it couldn't separate the apple flies from the hawthorn, as you can see by the mixture of red and blue. This means we know the dogwood flies are different on a genetic level, an important step forward in understanding the complicated Ragoulides system as a whole. What's more, with my additions, Entropy can be applied to nearly any other species on the planet. It can analyze fish to curb mercury pollution, birds to predict seasonal migrations, or even mosquitoes to help stop the spread of malaria. Thanks for watching, and I hope you've been able to see how computing is revolutionizing research in population genomics.